Welcome everyone, my name is Sebastian Bury and I'm a PhD student from the Institute of Dendrology of the Polish Academy of Sciences in Górnik in Poland. And I want to present the results of our studies, impact of Prunus serotina and Robinia pseudoacacia on temperate forest regeneration. That studies are part of the Marcin Tedeschi project, impact of invasive tree species on ecosystem services, plant biodiversity, carbon and nitrogen cycle, and climate regulation, funded by the National Science Center of Poland. Prunus serotina, commonly named white black cherry, and Robinia pseudoacacia, commonly named black locust, are among the most common invasive trees in Poland. Both origin from North America. As many of the invasive species, they can transform newly occupied ecosystem, contributing to the loss of biodiversity. The other consequences of their presence are economic losses connected with removing them from forests before artificial regeneration. And we can suppose that neophytes can affect natural forest regeneration, which is one of the most important components of forest sustainability and forest functioning. So the aim of our studies is finding the answer of what is the per capita effect of Prunus serotina and Robinia pseudoacacia on forest regeneration. And it should help us to better knowing the role of invasive tree species in forests and contributing to their better management. We hypothesize that both Prunus serotina and Robinia pseudoacacia will impact natural regeneration. These two species differ in nitrogen fixing strategies, and in Europe the range of biomass achieved by individuals of these two species differ from each other. So the quantitative gradients and ecosystem influences should be different. We also expect different impact of that species on particular stands and habitat variants. And the other neophytes should benefit from their presence according to the invasional meltdown hypothesis. The studies were conducted in Western Poland on the forest area managed by state forests in the three inspectorates, 𝙑𝙑𝙪𝙑𝙪, 𝘗𝘳𝙀𝘀𝙀𝙀, and 𝘱𝙀𝙀𝙀. And we established 72 circular plots, each of 500 square meters in size, in few variants. Variants like invader species, it was Prunus serotina or Robinia pseudoacacia. Habitat type, nutrient rich with oaks, quercus, and nutrient poor with pines, pinus. Stand age, medium or mature. And we selected plots in stands from 44 to 139 years old, and stands with different invasion levels, so we can distinguish control plots with 0 biomass percentage of invaders, and invaded stands with different abundance of studying neophytes. For Robinia, it was from 0.5 to 69%, and for Prunus serotina from 0.1 to 10%. And the distance between individual plots from individual variants was greater than 5 kilometers to avoid spatial autocorrelation. On each of the plots, we inventoried the entire natural regeneration up to 1 meter and 30 centimeter high. Seedlings were not included in further analysis. All specimens were counted on 4 circular systematically disturbed subplots. In order to determine the intensity of the invasion, we were made some stand structure measurements like diameter at breast height of all living trees on each of the plots and height of the 20% individuals of each species. And then we used biomass formula to calculate above ground biomass, including leaves, stem, and branches, according to the studies of Tedeschi and Jagodziński in 2021. All analyses were made in our software, and we used generalized linear mixed models to predict changes in the per capita impact on the abundance of generation, like summarized abundance of all woody species and summarized abundance of all alien species. We also analyzed separately for most frequent species like Acer pseudoplatanus, Carpinus betulus, Quercus petrea, and Prunus serotina. We also included in the models independent variables like stand age and habitat types. And we used forest inspectorate as a random variable. In order to study the species compositions of the regeneration, we use the canonical correspondence analysis with consideration of scent age, type of habitat, invasive tree species abundance, and species of invasive tree in the stand. According to the CCA model, per capita effect of invaders are significant. And you can see in this graph that blue dots group separately from the other colors of dots. These blue dots are plots with Robinia pseudoacacia in stand, while control plots, red dots, and plots with Prunus serotina, green dots, mixed with each other. And we can see that samplings like 𝙪𝙪𝙪𝙪𝙪𝙪𝙪𝙪, Corylus avellana, and for example Sambucus nigra goes with stands with Robinia. And species compositions are also well separated for nutrient-poor sites. On nutrient-rich sites, there are two interesting species, Cerasus avium, which is native for Poland, and Prunus terracifera, which is invasive. They are similar if we talk about phylogenesis, so we can suppose that in the future, the negative effect of Prunus terracifera can be harmful for Cerasus avium because of the niche similarities and possible competition for resources. Increasing abundance of Robinia pseudoacacia can affect natural regeneration according to the GLMM models. Acer pseudoplatanus, Prunus serotina, alien species, and total species gets benefit, but 𝘗𝘃𝑃 petrea are a loser. We didn't find significant result in Carpinus betulus regeneration. And the model predicted that increasing of Robinia biomass, about 25%, will cause the increasing of Acer pseudoplatanus individuals approximately by 4, Prunus serotina by 0,6, all alien by 1,5, and all woody species by 10, and decreasing of 𝘗𝘃𝑃 petrea by 12. In the stands with Prunus serotina, we found the same trends, but intensity is quite different. The proportion of Prunus serotina in all alien species are approximately 90%. And the model predicted that increasing of Prunus serotina from 0 to 3% will cause the increasing of Acer pseudoplatanus individuals approximately by 7, Prunus serotina by 7, all alien species by 8, and all woody species by 35. And decreasing of 𝘗𝘃𝑃 petrea by 30. It's changing a little bit when the invaders are growing from 3 to 7%. We can see the increasing of Acer pseudoplatanus individuals approximately by 26, Prunus serotina by 37, all alien species by 38, and all woody species by 63. And decreasing of 𝘗𝘃𝑃 petrea by 9,5. So we confirm that per capita effect matters and was different in the case of invader species and invader biomass range. We also found the similarities and differences between native species response on invader presence in stands. We also confirmed the invasional meltdown hypothesis. And as a part of our research, we plan to conduct a larger number of plots to diversify the quantitative gradient of invasion as much as possible. The context of climate changes are also important in our studies. Acer pseudoplatanus will be a winner of the climate changes, so invasive species like Robinia and Prunus serotina can intensify this process. From the other side, 𝘗𝘃𝑃 petrea may be retreat from Western Poland and wander east, so these two invasive species can intensify the negative impact of climate. So at the end, our studies are important for invasive species management in protected and managed forests. Thank you so much for your attention.